Good evening and welcome to your class in CPT 3 Fundamentals of Crop Science. Last time we discussed about climate change and um, there are videos to show you for additional information and also the recorded video of last time's discussion. So now we are going to talk about biotechnology. Its overview, concept, application, and status. So I just got this from CERTA, from my friend, faculty before in USEP. So in this presentation, you will see the historical development and how to define biotechnology, genetic engineering process, and its applications, global status, and biotech crops. So when we say biotechnology, what comes in your mind? Like, for you, what is your idea with biotechnology, Oliver? Nawala si Oliver. John Ray. Hello, John Ray.
Yes, ma'am.
For you, what is biotechnology?
Biotechnology, ma'am, is mga bagong, ma'am, mga new technology.
Yes, it's a new technology, of course. So when we say bio, what does it mean? It refers to all living things or all living organisms. So in terms of historical development, actually, they say that biotechnology is as old as civilization, no? From even when they say agriculture is also as old as civilization, like we we hunt for food, we look for food, and we domesticate food or crops and animals. So that's already a form of agriculture. So during the hunting and gathering pastoralism time, so the man or the Homo sapiens and his early ancestor existed as gatherers and hunters of food. So that time, it is where also the time when biotechnology agriculture started. So domestication of animals and selection of plants began about 8,000 before Christ. And Egyptians mastered the art of winemaking and baking leavened bread about 4,000 BC. So that's also a form of biotechnology. In 1760s, age of industrial revolution, the use of mechanical implement and machineries in agriculture and crop milling started. So, as early as 1760s. Meron na tayong mechanical implement. And on 1860, deliberate plant breeding, pollination of plants started. So, we already developed new varieties at this time. In 1960s to 1970s, the period of agriculture advance known as the Green Revolution was started. So we say Green Revolution because we already use high-yielding varieties and fertilizers, irrigation water, and pesticide to increase yields significantly and strengthen food security. So 1960 to 70s is what we call the period of Green Revolution, or we say agricultural advance. And that's because, again, we use of high-yielding varieties and so on. On 1953, Watson, Crick, Wilkins, and Franklin elucidated DNA structure. So it's the time they come to know about the DNA or the genetic material of all living organisms. In 1973, the discovery of the recombinant DNA technology by Cohen, which is known as the birth of modern biotechnology. So they say modern biotechnology before 1973, that's a form of biotechnology, but when we say using recombinant DNA in 1973, it is the modern biotechnology. And in 1983, after 10 years, the first genetic engineered product, we call it insulin, was produced. If you know insulin, it's the, yes? You have any question? What is insulin?
Insulin produce.
It's the substance that controls sugar content in the body, right? And it's for diabetic person. So in 1990, the first successful field trial of the genetic engineered BT corn, cotton pala, not corn, was a successful, first field successful in 1990. So we say, are you familiar with BT? BT corn, BT cotton, there's a lot of BT. So BT stands for Bacillus thuringiensis. It's kind of bacteria which when the insect eats that one, deteriorate and die. No, so that's why it's engineered inside the genetic makeup of cotton. So when the pests attack, so they get die. In 1990, the Human Genome Project was established. And this is in the case of human beings. In 1994, a product known as flavirsaver tomato is approved by the Food and Drug Administration, and it's the first biotech food crop to be released. In 1996, biotech crops are grown commercially already over nearly 1.7 million hectares and 4 million acres worldwide in Argentina, Australia, Canada, China, Mexico, and United States. And in 1997, the birth of the first cloned animal known as the sheep dolly. In the year 2000, biotech crops are grown on 44.2 million hectares already, about 109 million acres, in 13 countries. And the first complete map of the plant genome developed is in Arabidopsis thaliana. So it's the first plant wherein the complete genetic makeup was mapped or developed. We also have the golden rice, no? It was very popular about 5 years ago, but now there's no news on this one already. Announcement allows the technology to be available to the developing countries in hopes of improving health, undernourished people, and preventing some forms of blindness. So it's yellow color, that's why it's golden rice, and it's for vitamin A deficient. In 2009, as you can see on the figure, biotech crops are grown on a lot of countries already in 134 million hectares in 25 countries. So, and it's been increasing and increasing, no, up to 2009. So what are the scope of biotechnology? So does it include all living organisms, plants, animals, virus, and bacteria? John Ray, are you there?
Yes, ma'am.
Okay, so is biotechnology for all living organisms? Yes or no?
Yes, ma'am.
Okay, very good. So in fact, the genetic engineering of animals, which are mainly for livestock, is already even until now under development, no? To develop animal vaccines and biocontrol agents against plant pests and diseases. and also plant protoplast fusion, and even embryo transfer. So,
Saan meaning GE, ma'am?
genetically engineered.
Okay.
Kasi biotechnology as a whole is, as you have said, is the advanced technology for all living organisms. So it's also about genetic engineering is actually, uh, it's because biotechnology is so general. As I have mentioned earlier, it even started from hunting, gathering food at that time, no? When civilization was first started, so it also starts with that time. So but when you say genetic engineering, it's the more advanced part of biotechnology. So there are also genetically engineered crops and animals to improve microorganisms, and recombinant DNA for disease diagnostics, monoclonal antibody production, plant tissue culture is also another scope of biotechnology, as well as the fermentation and biofertilizers. So if you have heard biofertilizers, these are fertilizer with microorganisms. And fermentation are just example is cheese. It's they say cheese is actually the one of the oldest genetically engineered food. And beer is it's fermented, and even wine. And these are all scope of biotechnology. So what is biotechnology? Bio means life and technology means technique or procedure to develop new products. So if literally it's a technique or procedure to develop new products in all living organisms. So if we define biotechnology, it's just the use of living organisms or their parts to make modified products or improve plants, animals, and microorganisms. So it's also very complex discipline because it's interdisciplinary. So it involves biotechnology, involves genetics, microbiology, biochemistry, food technology, molecular biology, biochemical engineering, food science, breeding, and mechanical engineering. So what is this recombinant DNA or genetic engineering? It's a set of technologies that artificially move functional genes across species boundaries to produce novel organisms. So what are these examples of novel organisms? This is what I have mentioned earlier when the time that the first FDA approved genetic engineered tomato was released. So the BT cotton and the BT corn are some of these examples, no, of the recombinant DNA genetic engineered crops. So how does genetic engineering goes? So first, the gene is of interest, like the Bacillus thuringiensis. It is cloned and introduced into the cell, and the cell expresses the gene, and the proteins are produced. Then the trait is expressed, but that is not as easy like that before. If you have your genetic, do you have your um plant breeding subject already? Not yet.
Not yet, ma'am. Ay, plant breeding.
Yes, not yet.
Wala pa, ma'am.
Yes. So to develop a variety through natural breeding is actually takes like 20 years, no? But through genetic engineering, through DNA transcription, RNA translation into protein, and by the technology that we have now, it's just easy as like 5 to 10 years, no? With the traditional, compared to the traditional breeding system, which takes 20 years or more. So what are the requirements in genetic engineering? So you need a vector. So the carrier of that gene of interest. And the host cell, so where are you going, like the host cell, usually bacteria, wherein you want the gene, because it's easier in bacteria and other unicellular cells. And the procedure to introduce the gene into the cell. And you need time and research funds because it's very expensive and it needs a lot of equipment and chemicals to do that. And a clone gene of interest. So if you want bigger, like higher yield, something like that, so the genes responsible for that could be your gene of interest. So in crops, genetic engineering is precisely breeding with the precision and accuracy of genetic manipulation. Like, for example, why do we use genetic engineering for crop improvement wherein natural breeding occurs, right? So it's because we want the trait of interest which is not present in the crop germplasm. So we want a crop that yields higher, but is not susceptible to pests and diseases, right? So, but there are varieties, and most of the varieties of our crops are not as perfect as that. So we want it to be perfect. That's why we need genetic engineering. So sometimes trait is very difficult to improve using conventional methods, yung natural breeding. Like, have you seen how to breed a corn? Have you seen?
Wala pa, ma'am.
Wala pa. So usually ka ng, because corn needs, although it has it both have its own flower, and male and female flower. You know where is the female flower of the corn?
Ito, asan eh?
The tassel is right. Yes. And then yung kanyang cob, yung nasa baba is female. So, although complete flower siya, but then the 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 problem with corn, that is why siya, cross pollination siya. It doesn't have its self pollination, unlike most crops like pomelo, it's self pollination. But for complete flowers, I'm talking about complete flowers. But for corn, it has both the male and female flower, which are separate. But then they cannot self-pollinate because yung tassel before yung kanyang ovary ng corn ay nagmamature na before yung sperm ng tassel natatanggal. So usually cross-pollinated talaga ang corn. So
Ana may putlon, ana
really, can you share?
di ba na may putlon, ana?
Ang sa putlon?
Yung pag-pollinate ba?
Yang bungot?
Oo.
Okay, para mas madali ang kwan. So you have tried already?
Wala ma'am kanang na parang na mo na discuss ko niya tung sa kwan pa mi sa crop 1 sa una.
Okay.
Subject na mo crop 1.
Crop 1. What is your crop 1? Kung damin CPT3 is different sa inyong crop 1? Lahi ka na
Like ma'am. Sa kwan man to sa science ma'am to sa ah Mantapo ma'am.
Okay.
Pamilya ka na sa eskwelahan?
Yes. Yeah, so transfer day ka.
Opo ma'am.
Okay, so how
2 years naman may dito.
Not 2 years. So how would nakaani?
Dili ba nakalimtam siguro ma'am.
So sometimes like that, no, the trade is very difficult to improve using conventional methods. So another reason is that improving the trade conventioning will take a very long time. As I have said, it will take 20 to 30 years. So genetic engineering seems to be the answers that allows the transfer of a specific gene for a desirable trait. So overall, ito yung usually what happens in genetic engineering. So you choose your gene of interest, and these genes will be encoded or transferred into a cloning site or vector. We call it gene marker. And then inside that vector, there's genes construction. For example, agrobacterium mediated infection. So this particle bombarded, so and will be bombardment in shooting. So in this way, this is transferred through plant tissue culture. And then it is selected through process and regenerated, and then transferred in the greenhouse for selection and testing. So for field testing, those who who have this characteristic with the gene of interest are selected to make a new variety. So this is another way of genetic engineering through tissue culture with an infection of agrobacterium. So sometimes there are also other genetic engineering which uses very advanced, no, yung cell-to-cell culture at saka yung gene-to-gene culture. So other applications of biotechnology, as I have said, bioprocess technology, the production of essential primary metabolites like acetic acid, lactic acid, glycerol, and industrial useful enzyme lactases, and monoclonal antibodies, vaccines, and novel recombinant products. So vaccines, so we're creating now vaccines for the COVID-19 pandemic. So that's also another biotechnology. Environmental biotechnology includes waste treatment, pollution control, detection and monitoring environmental contaminants, removing toxic waste through bioremediation. So when we say bioremediation, it's the use of plants to remove polluted or toxic waste like lead, ano pa dyan, yung mga toxic na mga metals, no? Which are harmful to the environment. So there are like kangkong, be careful of kangkong because it is also a bioremediator. So usually it absorbs a lot of toxic chemicals. So you only eat kangkong when you know the area is not contaminated with toxic chemicals. So recovery of metals from the mining waste and low-grade ores, we call it phytoremediation accumulation. So phytoremediation naman is another way, like this recovery of metals from mining waste. So next is plant biotechnology or genetically engineered plants. So as I have mentioned earlier, the golden rice one, it is is for nutritional enrichment in oil or starch or essential amino acid. And it has improved resistance to insect pests and microbial diseases. So example for this is the BT corn or the Bacillus thuringiensis corn, kanina BT cotton, and the PRSV resistance, also virus for the one that is earlier mentioned in papaya. Another genetically engineered plant is this improve yield and stress tolerant, drought tolerant crops with aluminum and manganese tolerance for acidic soils, like the tomato, and improve post-harvest characteristic like the flower saver genetic manipulation for cut flower industry. Another plant biotechnology is the tissue culture technology. And this is very common and being used already in banana and orchids. Another one is biological control using the BT insect and the rhizobium, no nitrogen fixation, and also bioherbicides. So for animals, genetic engineering for transgenic animal, because this biotechnology is, although this subject is for crop production, so but then biotechnology is very general, so I just included some animals here for your additional knowledge. So we call it super mouse. It's the first genetic engineered animal with growth hormones in gene, and introduce economically important traits into commercial livestock, which improve quality of meat, egg production, wool quality, and quantity. So if you notice now, but it perhaps because of hormones, some of the breeds of chickens now easily, can be easily harvested because they just grow mature earlier and they grow bigger, So those are also, we can say that, part of biotechnology or genetic engineering. So we also have genetically engineered hormones and vaccines for bovine growth hormone called somatotropin, or BST. It's the first biotech-derived pharmaceutical commercialized for animal agriculture, and these are injected to cows for increased milk production, and a lot of vaccines for animals. For healthcare, another application of biotechnology, we have vaccines, as I have mentioned earlier, monoclonal antibodies for hepatitis B vaccine, and hopefully we have COVID vaccine already in the near future. Biopharmaceuticals like the insulin for diabetic persons, somatostatin as a growth hormone, interferons for cancer and virus diseases. And we also have this called gene therapy, treatment of genetic disorders of disease cells. So this is highly complex and its potential could yet be realized in the near future. So this is still under research. So that's all for the applications of biotechnology. So another report for the status of biotechnology or genetically modified crops. So as for up to 2009, it has been increasing in terms of area and in terms of developing countries. Mostly genetically or biotech crops have been produced in the developing countries actually. And in terms of crops, genetically modified or biotech soybean is highest among maize, cotton, and canola, and it's everything is increasing, no? And in terms of like a trait, herbicide tolerance, insect resistance, and herb tolerance, insect resistance, here it's also, the usage is also increasing, as you can see. And another one is versus conventional and biotech. Now, more in soybeans, there are more biotech soybean now compared to conventional. For cotton, it's almost equal, but for maize, no, for corn, so more of conventional pa rin compared to biotech, no. And for canola, if you know canola, it's the canola oil, no. So more of, if you use canola oil, so that's more of conventional compared to biotech. So by trait naman, kasi meron din tayong tinatawag na RR corn and BT corn. RR corn is Roundup Ready corn. So it's another biotech crop. So as you can see, more of the BT corn, the blue ones, and the RR corn than the stock. And here, as the years goes by, so more on the both BT and RR corn now. So the adoption of BT corn in the Philippines, it's more of Luzon and Mindanao than Visayas. So in the Philippines naman, the adoption of this 5 genetically modified or biotech crops, the BTR, the BT, the Roundup Ready, and both. So it has been widely already introduced and used in the Philippines as of now. So through the Philippine initiatives of biotechnology, we have the papaya resistant virus, papaya, the BT eggplant, the delayed ripening papaya, the golden rice, the BT cotton, and virus-resistant sweet potato. These are just some of the initiatives on biotechnology in the Philippines. So through the Institute of Plant Breeding, the Philippine Rice Research Institute, the Cotton Development Authority, and also VSU and UPLP. So that's all for that status of biotechnology. So I also have another slide for more information on biotechnology. So I'll just introduce you because we just have 7 minutes left, because this is just supposed to be for 30 to 45 minutes meeting. So if you have questions, you may raise your question now about biotechnology or any of the topics that we are going to have. Oliver and John Ray, do you have any questions or concerns regarding biotechnology?
No, ma'am.
Kuan ma'am, usay kalayan ng kuan propagation og plant breeding. Propagation of plant breeding.
Well, plant propagation is the general term which include plant breeding. Because when you say plant propagation, it's the way wherein you propagate plants through sexual and asexual way. So plant breeding is the sexual way because you make use of the reproductive organs, no, for sexual for plant breeding. So
Ah,
there's also
so
plant breeding that makes use of tissue culture. So usually they are used interchangeably. So plant
pero asa mas better niya ma'am?
huh?
Asa mas better sa duwa kung kanang ginagamit sa farm ba?
Plant breeding actually is very specific, and plant
Ah,
propagation is general. So there are a lot of types of plant propagation. Anyway, if we will be talking more of plant propagation here, no, in Unit 4.
ah,
Now you will be introduced to different types of propagation, including yung mga grafting, budding,
body.
yes.
Dili.
So, some more questions.
No ma'am.
So, this is another definition and explanation on biotechnology also, but this is also a note from my classmate before in UPLB. So as you can see here, she has a lot of definition of biotechnology. So you can choose your own definition actually, so long that you know what is bio and you know what is technology. No? So even naka-stages pa diri ang biotechnology. So last time, the first handout, I mean the material that I have discussed talked about the history. But when you say stages, it could be ancient, classical, or modern. No? So we will discuss further on this on the next meeting, because this will explain in details on how to manipulate the DNA talaga. Although we will not be seeing that in actual, because Genetic engineering is actually an abstract form of science. Like you don't really see in your naked eye, you cannot see a gene, no? In fact, if you have your biology before, nag-sequencing mo og gene sequencing
Sa
before.
genes mo.
Yes.
Na, sa DNR mo to.
Yeah, kaning mga mga UCAG ana. Making of proteins.
Na protein. Ano siya, say tawag ano yung
Sequencing gani.
Oh.
I also handles NatSci 22 now, and we will be discussing that, no? So, but if you have come across with that before, so it would be easy for you to understand this next topics, because these are more on the recombinant DNA technology. So this is very specific, no? And you can also see here examples like the cheese production. So you will say, are you in favor of this technology or not? There has been a,
Favor
there has been a lot of debate already until now on whether pro GMO baka, genetically modified organism, or not, no? So how about you, Oliver and January? Are you pro GMO or not?
GMO.
Yeah,
Sarang GMO.
genetically modified organism or genetically engineered crops and animals. Hi, Jessie.
GM zero.
Yes.
GMO mga ma'am.
Huh?
Wala si ma'am. GMO.
Pro GMO mo.
Yes ma'am. Sa amo ang application sa amo ang animals.
Mm-hmm. So, even cheese, no? Kung kaon ba mo cheese? It's my
Yes ma'am.
Yes, so we've been eating cheese and it's already part of our culture since Spaniards' time, maybe. So we cannot say that we don't want GMO, no? Kay nagkakaon na tag cheese, nagainom tag beer, nagainom tag wine, and so on. Jessie? So anytime, yes, anytime the video will be stopped because we have limited time only. So actually, yes, mabilis ko lang na-discuss yung first material on biotechnology. So in that material, we define biotechnology, what is biotechnology, the history, the status, the examples. So this material will be discussed naman in detail next meetings. So you just
Ano sa ma'am?
you just go back to the video. I'm